Hi there. I really apologize for not appearing in person, but I'm unable to travel at the moment for medical reasons. And I also want to apologize for changing the focus of this presentation slightly. Today I'm going to talk about the role of DNA repair gene mutations, including truncating PPO&D mutations on acute neuropsychiatric decompensation, a situation that occurs in a small minority of kids with Janssen-DeVries syndrome. I have no financial disclosures. So first, a review of DNA damage response, which I think many of you already know. A central player in this is p53, a protein that is activated when DNA damage occurs. It's activated through the action of these kinases, ATM, ATR, CHEK1, and CHEK2, which add a phosphate group to p53, causing its activation. DNA damage occurs in cells all the time when a cell undergoes cell division in response to infection, in response to other types of stress. DNA damage always occurs, and we have a very, very sophisticated DNA repair processes, many different pathways that will repair the DNA damage depending on the type of damage that occurs. And p53 is a major factor. Now, PPOD is a phosphatase, which removes phosphate groups, and it kind of acts as a brake to activation of p53. So it's a negative regulator of p53. And every time you have a situation in cells, whether it's cell growth or clotting or DNA repair, all these activation phenomena have to have a brake that limits the action. Too much cell growth is no good. Too much clotting is no good. Too much DNA repair might be problematic as well. So this is part of the yin-yang homeostasis process that occurs in physiology. And in patients with truncating exon 5 and exon 6 mutations, as seen in Janssen-DeVries syndrome, there's an increase in phosphatase activity. Now, PPMD also dephosphorylates other proteins. One of them is p38 MAP kinase, which is a protein that's involved in dendritic spine function. Dendritic spines are these structures that sit on neurons. This is a neuron. These are these projections called dendrites. And sitting on dendrites are these dendritic spines. Here's a close-up view of them. And this is where learning and memory occurs. Spines are very dynamic structures. They change all the time. So listening to this talk, listening to other talks is affecting these spines in various parts of the brain. And this is basically the fundamental process through which synaptic plasticity occurs. And this is the change in synaptic architecture due to learning and memory. Now, these spines have hundreds of different proteins, and many of the genes that code for these proteins are well-known players in autism, neurodevelopmental disorders or NDDs, and schizophrenia. So this actually constitutes the most common cause of these conditions, mutations in the genes that affect these spines, as well as mutations that affect these presynaptic neurons that synapse onto these spines. PPOND also dephosphorylates mTOR, which is a major signaling pathway involved in regulating cell growth. And this pathway is dysfunctional in several NDDs, most notably tuberous sclerosis and PTEN-associated NDD. These studies were done in mouse knockouts. That is, these mice have no PPOND genes that are functional. This is the classic knockout model for studying all types of disorders, from cancer to NDDs. This is not the best model system for Janssen-DeVries syndrome because the mutations hit in PPOND are gain of function. They're not loss of function. And hopefully, Dr. Vivian will have something to say or some updates about his mouse model, which would be the most appropriate way to study either synaptic spine function or the effect of PPMD on this protein and on P38F kinase and mTOR and other proteins. So the question I'm going to address is whether defective DNA repair per se is involved or playing a role in any of the clinical manifestations seen in Janssen-DeVries syndrome. So here's a review of p53 phosphorylation. Again, it's activated by DNA damage. This activates these kinases, ATM, ATR, check1, and check2. At the same time, PPMD is activated. So while this is carrying out its DNA repair function, PPMD is acting as a brake. It's dephosphorylating p53 as well as several other proteins, two of which are not directly involved in p53 phosphorylation, but are involved in DNA repair. This is gamma H2AX and 53BP1 proteins that will come up a little bit later in this talk. So we are interested not only in PPMD, but other genes that are involved in DNA repair and the effect that might have on acute and subacute neuropsychiatric decompensation that leads to two overlapping conditions called PANS and regression, which we've found so far in only 3 Janssen-DeVries cases. Now, PANS is a clinical diagnosis based on criteria established by experts in the field. Kids have to have the acute onset of OCD and or restricted food intake, plus 2 or more of these symptoms: anxiety, emotional ability, irritability, aggression, very aggressive behavior, severely oppositional behavior, and developmental regression. These kids begin to perform poorly in school. They forget how to do math. They forget how to draw. So it's a very, very disturbing set of symptoms. They also have some loss of urinary control and increased urinary frequency. And most kids, instead of having 2, which is the minimum criteria, which are the minimum criteria for having a PANS diagnosis, including the abrupt onset of OCD or restricted eating, they have more than 4 or 5. And what's characteristic is the explosion of symptoms that occurs in these kids. It happens sometimes overnight. It can happen over the course of a couple of days. It's very, very different from OCD. But because there's no objective test for PANS, the diagnosis remains somewhat controversial. That's a whole other lecture I can give. So PANS episodes are triggered by infections or non-infectious stressors, like metabolic stress, and a very unique subtype of PANS is PANDAS, which is triggered by group A beta hemolytic strep. This is characterized more by OCD and tics. And it can occur in a kid that has no history at all. It can occur in a child with pre-existing autism or any NDD. And it's found in kids who have an underlying autoimmune problem or an immune deficiency, especially CVID, common variable immunodeficiency. And as I said, it's a clinical diagnosis based on the criteria that I showed you on the previous slide. And it's recognized by the NIH as a rare disease, but it's not in DSM-5, the diagnostic manual psychiatrists use as a guide and to teach residents. Consequently, most residents in the United States and in the UK and the EU really don't have any good knowledge of this condition, which makes it very frustrating for parents. But those of us in the field think that PANS is an autoimmune or autoinflammatory disorder. I don't have time to go into that distinction, but you can view them kind of under the same umbrella. And part of that belief is based, first of all, on the fact that it almost always comes after infections, and the fact that many cases, many kids with PANS respond to an inhibition of the immune system or some other type of immunomodulation, most notably IVIG, which is intravenous immunoglobulin. They respond to steroids and the B-cell inhibitor rituximab. Not every kid responds, but enough respond to make these the case for treatment in some cases. They also respond to antibiotics. Now, regression in autism and NDDs refers to the loss of previously acquired skills, and can be in any of the autism domains. And it can occur at any age, and it's quite common, maybe under-recognized, but quite common. And there are many, many different types. In one type, most notably Rett syndrome, this is part of the natural evolution of these conditions. In Rett syndrome, these kids meet their milestones for the first 18 to 24 months. And then, like clockwork, they begin to have a severe decline. And you see the exact same thing in the mouse model of Rett syndrome. In other cases, it can occur at any time, sometimes following an infectious disease. And this too, regression too, is believed by many to have an autoimmune basis. And just like in PANS, some of these cases respond to immunomodulation. Now, our first foray into the genetics of PANS occurred a couple of years ago. We found these genes, mutations in these genes in 21 cases. And most notably for this audience, one of the cases, case 3, has Janssen-DeVries syndrome. Now, I wasn't looking at this. I knew this of this young man who, as a child, was diagnosed with PANDAS, and he was not known to have Janssen-DeVries syndrome until maybe 6 years later, when a workup for an NDD found a classic truncating mutation in exon 6. What I found most intriguing, all these genes have a story and I have no time to go into it, but the most important element related to this talk is the fact that 3 of the genes have an effect on DNA repair. And the most interesting combo is the Janssen-DeVries mutation in PPMD, and this loss of function mutation in CHEK2, which you might recall is a p53 kinase. So you have a loss of expression of a kinase and the increased expression of a phosphatase, and both would be expected to lead to an impaired or reduced p53 phosphorylation. This idea was cemented in our next study, where we found 2 other cases of Janssen-DeVries syndrome who had either PANS or acute regression, or in case 1, both, as well as mutations in a couple of families with ATM and ATR mutations. The ATM mutations are the most substantial, the clear pathogenic mutations. There was one young woman with a loss of function mutation, and here you have a family of 4 kids, all of whom have either autism, regression, and/or PANS. So this kind of cemented the notion of DNA genes being a problem or a cause of acute neuropsychiatric decompensation. And we found families and individuals with mutations in members of the Fanconi anemia complex, which have a role in certain types of DNA repair and actually interact with the p53 pathway. It's hard to see on this slide. Now, these are very, very rare conditions, especially in Janssen-DeVries syndrome, DeVries syndrome. And one of the reasons for this may be the fact that other mutations need to be found or need to occur in these individuals, suggested in many cases, but maybe not all, in many cases of a 2-hit hypothesis. And the one in case 1, the Janssen-DeVries patient in case 1, he has a de novo pathogenic mutation in this gene that codes for casein kinase alpha-1, casein kinase 1 alpha-1, which is a kinase that has effects on synaptic function, on p53, and a DNA repair, and it has a suppressive effect on the innate immune system. So loss of function can lead to autoimmune problems. So the combination of that mutation plus the truncating mutation PPOD is what we believe sent him over the edge in terms of neuropsychiatric decompensation. And ATM, just like the CHEK2, is a kinase. So it is acting kind of in concert with the PPOD mutations, at least when it comes to trying to understand what is going on in these patients' immune cells. Now, DNA repair problems can lead to genomic instability. In some cases, in non-immune cells, this can actually lead to cancerous transformation. In immune cells, this can lead to activation of the C-gasting pathway. C-gasting is a pathway activated by viral infections and other types of infectious disease stressors, and it leads to the production of type 1 interferons, which are the major players in the antiviral response. So when you get a COVID infection, the type 1 interferons go up, although COVID infections, patients with COVID has outsmarted the immune system, and it inhibits type 1 interferon production. In any event, we believe that the combination of an infection activating this pathway plus an underlying DNA repair problem is leading to overactivation of C-gasting, leading to problems in the cell, leading to an autoimmune or autoinflammatory process. And C-gasting has been implicated in lupus and in various NDDs. It's a very hot topic. You go to PubMed and just type in C-gasting and autoimmune or neurodegenerative, you'll see many, many different citations. Then in addition to type 1 interferons, activation by these pathogens can also lead to activation of AIM2 inflammasomes, which lead to the activation of these pro-inflammatory cytokines, interleukin-1 beta and interleukin-18. So there's an overall pro-inflammatory activation response that is exacerbated by these DNA repair problems. Now, our two studies have been, in my view, replicated by two studies that came out in the last few months. One of them is in Down syndrome regression, which, unlike Janssen disease syndrome, is rather common, found in 15% of cases. And in a genetic analysis done on those cases, 8 de novo mutations were found in 41 cases, and 4 of them were found to affect type 1 interferon. And of those 4, 3 of them are connected with DNA repair. Then another study came out looking at cases very similar to the ones we look at, which is PANS and regression, and they found 8 de novo mutations in genes that affect chromatin, which is basically which are genes that affect gene expression and various other functions of chromosomes. And of those 8, 5 of them are DNA repair genes, will have a major effect on DNA repair. It's not necessarily DNA repair primarily, but not necessarily primarily being DNA repair being the major problem. I have these 2 genes highlighted, KENS-L1 and EP300, because we have also found mutations in those genes in PANS cases. And in the case of EP300, that will appear in a paper I'm currently preparing, that in which we identified 15 additional DNA repair genes. So now the question is, what is DNA repair doing in cells relevant to behavior or to autoimmunity or to inflammation, which would be peripheral immune cells, which we are not studying. We don't have access to patients that easily. Instead, we're using our iPS cell model to look at the effects of DNA repair on neurons and microglia containing Janssen disease mutations. Most of you know that I've created a bunch of lines. This is actually only a partial list. These lines are made from blood cells, we isolate these stem cells, which are committed to producing red blood cells and white blood cells and platelets. And those cells are turned into iPS cells using these factors derived from the Nobel Prize winning discoveries of Shinya Yamanaka, a Japanese investigator who discovered how to make iPS cells. iPS cells have the unique property of being able to be turned into or differentiate into basically any cell type in the body. Gut cells, kidney cells, heart cells, skeletal muscle, and for our purposes, microglia, to the brain's immune cells, and neurons. In particular, glutamatergic neurons, which are the major excitatory neurons of the brain, implicated in NDBs and autism and schizophrenia. And microglia also have a major role in regulating synaptic function. They are regulators of synaptic plasticity, bringing home the concept of defects in these cells contributing to problems, as I showed in synaptic architecture, as I showed on a previous slide in the mouse knockouts. So we look at control cells, we look at cells containing truncating mutations, and we study the effect of DNA damage. But here's one experiment we did in neurons exposed to this drug called etoposide, which causes DNA damage. And we noticed right off the bat that neurons have some kind of underlying activation of p53 by phosphorylation because lots of phospho-p53. And this is due, I think, to our culture conditions, which are creating a kind of oxidative stress and DNA damage that constantly requires DNA repair. And there's a significant difference in the baseline status of phosphorylated p53 in controls versus Janssen disease samples. There's a decrease in phospho-p53. Following treatment with etoposide, we see activation at 4 hours, both an increase in p53 phosphorylation in both the control cells and in the Janssen disease cells. So there is some p53 phosphorylation, but there's a significant difference in the degree of phosphorylation in those cells in controls versus the PBOND samples. My point is probably not working, I'm afraid. But so it's this third pair, this black and the gray bar. These black and gray bars at the end. The control sample, the baseline samples before etoposide is the first set of black and gray bars. Here's a more visually striking example of this. These are microglia exposed to ionizing radiation, which induces DNA damage. The DAPI stain just stains nuclei, showing cells. And on the left, we have baseline activation of gamma H2AX and 53BP1, which you recall are proteins involved in the DNA repair response. There's very little baseline activity, but when we expose the cells to radiation, we see a robust activation of H2AX in the control samples seen on the top, on the right, in both H2AX and 53BP1, but it's a much more tepid response in the PBOND truncating samples seen in the bottom panels. And we see the same thing in neuroprogenitor cells exposed to ionizing radiation, a robust activation of both H2AX and 53BP1 in the control cells, and a very modest activation in the PBOND truncating samples. So we are pretty confident that in Janssen disease neurons and microglia, DNA damage is having a negative impact on the DNA repair response. Now the question is, what effect is that having on the cells? And here we are at the very beginning of our studies, These are very hard to do in iPS-derived cells because these cells take a long time to grow. They're very, very expensive to grow and maintain. And we are working on assays to study mitochondrial function, and as well as getting good assays for looking at cytoplasmic DNA and seed-gasting, which have given us a lot of problems. So we're still in trying to figure that out. Proteomics and RNA sequencing, we are carrying out. We know how to do that quite easily, but we don't have any results yet. One intriguing preliminary experiment is a cytokine release assay we carried out on microglia. So like cells in the immune system in the periphery, when these cells are exposed to either infections or DNA damage or some other kind of stress, they release cytokines and chemokines, which are these small hormone-like substances that are very important in controlling infectious diseases. So they activate the part of the immune system needed to control certain types of infections. And we've already done this quite extensively in microglia exposed to drugs that mimic a bacterial infection. I'm not going to show that here, but here's our first experiment looking at cytokine release following DNA damage. And I mean, it's one versus one, so it's really kind of meaningless. But what we see is a very intriguing result. We see a twofold increase in the in CCL2, interleukin-8, interleukin-13, and interleukin-18, which are pro-inflammatory cytokines and chemokines, and a similar degree of decrease in CXCL12 and interleukin-1RA, which are chemokines and other immune modulators that have an anti-inflammatory property. So the combination of two is suggestive of a pro-inflammatory process in microglia containing truncating PPL&D mutations. And all these cytokines and chemokines and immune modulators like IL-1RA, these have all been implicated in neuroinflammatory disorders and in neurodegeneration. So I think we're on the right track. So to summarize, PANS, susceptibility and regression in autism and NDDs are associated with mutations in genes that affect DNA repair, as well as many others, which would be 2 or 3 other seminars. And a very small subgroup of kids and adolescents with genetic degrees are susceptible. We've only found 3 so far. And it's, as I mentioned earlier, it's rare because it most likely occurs in the context of other genetic variants occurring in combination with the truncating mutation. And preliminary data show that DNA damage might affect the release of pro and anti-inflammatory cytokines and chemokines. Now, faulty DNA repair can have many, many different effects on the cell. It can affect gene expression. It can affect mitochondrial and lysosomal function. And there's a mountain of research that needs to be carried out, and we're starting to do those. As I mentioned earlier, we're starting to do those experiments now. So it'll take quite some time to go from the DNA repair defect that we clearly established in neurons and microglia in these truncating in these M1D mutated samples to what is actually happening downstream. And I would like to acknowledge my coworkers, especially the 3 people in my lab who do most of the research. And I want to acknowledge funding from the Janssen DeVries Foundation and other private organizations, one of which is very directly related to Janssen DeVries syndrome, which is the Ryan Stern IPSL research fund that we have. I have one pilot grant from the NIH that funded some of my DNA sequencing studies. And the families have been very, very helpful in me establishing my hypothesis. I don't see these patients personally, And almost everything I've learned about PANS and regression, I've learned from the parents of the 100 or so somewhat cases that I've seen by Zoom. So I really appreciate their help. And again, I apologize for not being there. If you have any questions, you can send them to Jensen Freeze Foundation. They will forward the questions to me, and I'll be glad to respond. So again, thank you very much, and I hope the conference goes very, very well.